After bobtail squid adults have been removed from their tanks and anesthetized with a 2% ethanol-filtered seawater solution, they are bled through the cephalic blood vessel with a 26.5 gauge syringe. The extracted hemolymph appears blue due to the presence of hemocyanin. The hemocytes are diluted to a desired concentration and aliquoted into an 8-chambered cover slip. Once the hemocytes have adhered to the cover slip, they are stained with a cell tracker orange, a fluorescent dye, and challenged with GFP-labeled bacteria, which can be visualized on a fluorescent microscope.
Hi, I'm Spencer Nyholm in the Department of Molecular and Cell Biology at the University of Connecticut.
And I'm Andrew Collins from the Nyholm Lab.
Today we will show you a procedure for extracting hemolymph, or blood, from the Hawaiian Bobtail Squid Euprymna scolopes. Now, bobtail squid are nocturnal animals found off the coast of Hawaii near coral reefs, and they use the light produced by their symbionts to camouflage themselves at night when they're out hunting for their favorite food, which is shrimp. Now, we use this association in the lab to study how beneficial bacteria colonize animal tissues.
We use this procedure in our laboratory to study interactions between components of the innate immune system of the host and its bioluminescent bacterial partner, Vibrio fischeri. So let's get started.
Before extracting hemolymph, anesthetize one adult squid in a 1-liter beaker containing a freshly prepared solution of 2% ethanol in 500 milliliters of filtered sterilized artificial seawater. After 10 minutes, the squid will cease swimming and will not actively respond to touch. However, continued respiration, as indicated by movement of the mantle, and chromatophore activity, indicating activity of the pigment cells, should still be observed. Pour some of the sterile seawater ethanol solution from the beaker into a standard wax dissection tray and submerge the squid in the tray with the ventral side up. Using one standard 200 microliter pipette tip, pull back the funnel and mantle to expose the main cephalic blood vessel located between the two eyes. Use a sterile syringe with a 26.5 gauge needle to puncture the cephalic blood vessel and withdraw between 50 to 100 microliters of hemolymph, or 10 to 20 microliters if the animal will serve as a donor multiple times. Place the hemolymph in a sterile 1.5 milliliter tube on ice. Note that if this step is done correctly, the squid blood should appear dark blue. Replace the animal in a normal seawater tank to revive. Spin the hemolymph in a microfuge at room temperature to pellet the hemocytes, and pipette off the supernatant. Resuspend the hemocytes in 500 microliters of squid ringer solution to wash, then spin as before to pellet the cells and remove the supernatant. Finally, resuspend the hemocytes in squid ringer solution. Determine cell concentration by hemocytometer, then add approximately 2,000 cells to each chamber of a glass cover slip. Allow the cells to adhere to the glass for approximately 10 minutes. At this density, the hemocytes should form a uniform monolayer on the glass surface. Keep the hemocytes on ice while preparing bacteria. In an orbital shaker at 28 degrees Celsius, grow one or more strains of Vibrio bacteria containing a green fluorescent reporter gene to mid-log phase in seawater tryptone medium. Using a spectrophotometer, measure the optical density of the culture at A600 to determine cell density. In a centrifuge, pellet the bacteria. Discard the supernatant and resuspend the pellet in 1 milliliter of squid ringer solution. Add bacteria to the hemocytes in the chambered glass slides with 100,000 bacteria per chamber. Incubate the slides at room temperature for 1 hour to achieve maximum binding. Add cell tracker orange at a final concentration of 0.005% to stain the hemocyte cytoplasm. Incubate for 30 minutes, and then wash with 150 microliters of fresh squid ringer solution. At a confocal microscope, view the stained hemocytes and associated bacteria. Enumerate the bacteria over the entire surface of the hemocyte. Now we'll show you some representative images of hemocytes challenged with the GFP-labeled non-symbiotic bacterium Vibrio harvii and the natural squid symbiont Vibrio fischeri. Here is an image of a hemocyte that was not challenged with any bacteria. The cell appears red and has become activated, so you can see its pseudopodia-like extensions. When the cell is challenged with the bacterial symbiont Vibrio fischeri, a few bacteria are seen binding to the surface of the cell. However, when the non-symbiotic bacterium Vibrio harvii is exposed to the hemocytes, significantly more bacteria are bound.
We've just shown you how to extract hemolymph from a bobtail squid. isolate the hemocytes, and use them in experiments to characterize bacterial adhesion to these squid cells. This protocol could be modified for immunocytochemistry by fixing the hemocytes after they adhere to the cover slip surface. RNA, DNA, and proteins may also be extracted once the hemocytes have been obtained from the animal. So that's it. Thanks for watching, and good luck with your experiments.